Good morning to everybody in the audience. Good morning. Hereby I open this academic ceremony in which Ms. Eline Berends will defend her academic thesis entitled Methylglyoxal and the Brain Microcirculation, The Source Matters. May I invite you to present a summary of your study and the conclusion of your thesis in the next 15 minutes. I give you the word.
Dear Prorector, dear members of the Corona, dear colleagues, friends and family. In the next 15 minutes, I'll be sharing with you what I've done in the last 4.5 years, resulting in the thesis Methylglyoxal and the Brain Microcirculation, The Source Matters. And I'll be doing this by breaking it down for you. I'll first let you know what the brain microcirculation is. I'll explain to you then what methylglyoxal is. And lastly, I hope by the end of this talk, I can convince you that in fact, the source matters. And I can start off with introducing my favorite organ to you, the brain. It's incredibly interesting because it makes us who we are, arguably also the most important organ of the body. It thinks, therefore we are. But it's also important in a lot of other functions. Think about movement and speech, but it also tells us when we need to sleep or when we need to eat. It makes us feel all these different kinds of emotions important for our survival. And lastly, we shouldn't forget, it also plays an important role in memory. So if our brain doesn't work properly, we one by one lose all these functions and we also lose ourselves. So how do we make sure that our brain keeps working properly? Well, we only have 15 minutes and I think it takes days to discuss everything we need for our brain to work properly. So I want to get back to down to the actual root of what is most important for our brain. Our brain is very functional, so it needs, Our brain is very functional, so it actually needs a lot of building blocks. Think about proteins and lipids. It needs a lot of oxygen and energy to function. And to get all of this in the brain, it needs to be supplied, which is done through the actual blood vessels. And the small blood vessels that do the delivering of all these important things is what we refer to as the brain microcirculation. And the brain microcirculation is highly specialized. It's really good, for instance, at regulating blood flow, which means that it can increase or decrease the blood flow by making the blood vessels larger or smaller, depending on what region of the brain needs most of the essentials. Another very unique feature of the blood-brain or of the brain microcirculation is the blood-brain barrier. And the blood-brain barrier is a multicellular complex structure that works together to form a literal barrier between the blood and the brain. And for this, I would like to use the metaphor of the wall around a castle. The wall around the castle really also protects what's coming in. It's really good at regulating what comes into the brain and what doesn't. And these features of the brain microcirculation are incredibly important. And if these things don't really work well, so for instance, a reduced regulation of the blood flow, a leakage of the barrier, or an obstruction of the blood flow, we can actually have major consequences for the brain. It could even lead to memory loss. Now, the collective term that we use to refer to the disease of the vessels and its consequences is what we call cerebral small vessel disease. And cerebral small vessel disease is actually incredibly common, and in particularly in elderly people. Therefore, age is also the highest risk factor in the development of cerebral small vessel disease. Another major risk factor for cerebral small vessel disease is diabetes. And for those of you who don't know, diabetes is a disease in which there is an increase in glucose in the circulation. But why are actually these factors such a big risk factor for cerebral small vessel disease? And I wouldn't, of course, be here if we didn't have a suspect. And our suspect brings me to the second part of my talk, methylglyoxal. Methylglyoxal is a very small and very reactive molecule. And what it can actually do is it can actually bind to our building blocks in the body, like proteins. And this changes the shape a little bit of these proteins. And you can imagine, like here in these building blocks, if we have methylglyoxal or a small molecule binding, it changes the shape a little bit, losing its functionality. And these bound proteins, these slightly changed proteins, are what we refer to as advanced glycation end products. And a lot of these advanced glycation end products could actually lead to disease. And why do we think methylglyoxal is actually, or why is it our main suspect and where does it come from? The body uses a lot of glucose to generate energy. And besides only creating energy, every once in a while, there is a little byproduct formed named methylglyoxal. And if you're healthy, that's no problem because we have a system to actually clean this up, the glyoxylase system. But you can imagine that, of course, if you have diabetes, it's not just a little bit of glucose in your blood, it's actually a lot. And there is still this generation of energy. And there's also the formation of methylglyoxal. But because there's so much glucose metabolized, we have a lot of generation of methylglyoxal. And the body can still clean it up a little bit, but this system gets overloaded over time, and it also reduces with age. So we can't really get rid of all this methylglyoxal. And this will then lead to this binding to these proteins, to advanced glycation end products, leading to disease. And this is what we think might be happening in the blood vessels in the brain. And to test this, we asked ourselves the question, does methylglyoxal affect the brain microcirculation? The first thing that we did to try to investigate this is we took a dive into the literature. What do we already know about methylglyoxal in the brain vessels? What we actually found is that methylglyoxal might possibly form a threat to the brain by damaging the blood-brain barrier. But a lot of these studies only looked into separate elements of the blood-brain barrier at the time and not the blood-brain barrier as a whole. So if you get back to this metaphor of the blood-brain barrier being a castle wall, it would be like looking at all the separate elements of this castle wall. So looking separately at the gate, the wall, the defense system, or the lookout towers. And the effect of all these different elements doesn't tell you anything about what is happening to the entire wall. The same for the blood barrier. Looking into one cell type doesn't tell you anything about what is happening to the whole construct together. So this is what we wanted to do. And then for this, we had to use an animal model, and we had to resort to the use of mice. And we actually used two groups of mice. And what we did to test the effect of methylglyoxal on this whole system is we gave one group of mice regular drinking water, and another group we gave drinking water with a very high concentration of methylglyoxal. And we did this for a longer period of time. And after this, we could collect the blood, and in the blood plasma, we could measure how much methylglyoxal is in there. And what we find is that if you put more methylglyoxal in the drinking water, the high concentration, there is also a lot of methylglyoxal in the blood plasma. So we have more methylglyoxal in the blood plasma, but what does this actually do to the small blood vessels in the brain? So we looked into the leakage of the blood-brain barrier. We looked if there was maybe any effect on the blood flow regulation. We looked for signs of inflammation, and we actually looked into what the effect on memory is. And I can keep it quite short because here we actually didn't find any effects. We didn't see any differences between the two groups. So that was a bit to a surprise. But as you might remember in the beginning, I explained to you how methylglyoxal is mainly formed from glucose. So we wanted to see what happens actually if we increase the formation of methylglyoxal by increasing glucose. So again, we had to use an animal model, and we used mice, and we had this healthy control. We had another group in which we increased glucose in the blood by inducing diabetes, which would theoretically lead to an increase in the formation of methylglyoxal. Then we had also a third group in which we also increased glucose and by inducing diabetes. But we also made sure that there was a genetic mutation overexpressing this detoxification system. So there is both an increase in formation, but also an increase in detoxification, which in the end should lead to less methylglyoxal. And these three groups, we again investigated what is the effect on the leakage of the blood-brain barrier? Is there any effect on the blood flow? And does this have any consequences for memory? And what we here found is that we didn't actually find an effect of methylglyoxal or the overexpression of the detoxification. We didn't find any effect on the leakage of the blood-brain barrier. There was no more increase in leakages. What we did, however, find is that the mice who had more methylglyoxal formed because they were diabetic had a slower neurovascular coupling response or a slower regulation of the blood flow. And this was improved by the overexpression of this detoxification system by glyoxylase. We also saw that the diabetic mice had worse cognitive function or worse memory, which was then again rescued by the glyoxylase. So there seems to be actually an effect, a negative effect of more methylglyoxal that can be rescued by getting rid of the methylglyoxal. But this is all in mice. And then you might wonder, what about humans? And we can, of course, give humans diabetes on purpose or genetically modify them to increase this detoxification for very obvious reasons. But what we can do is we can actually look into what is happening in the population and just measure what's going on there. So that's what we did. And the population that we used for this was the population of the south of Limburg, which is a whole study set up called the Maastricht study. And within the Maastricht study, not the whole population, of course, was measured because that would be impossible, but we used a representative sample of the population in which we can get an idea of what's going on in the entire population. And in this sample, the participants in this sample, a lot of things were measured. So think about, for instance, we know or have information about the age, about body weight, medication use, and disease. But what is interesting for us is there's actually blood samples of these participants in which methylglyoxal could be measured. Another thing is that they underwent MRI, and on MRI we can actually see if we can find signs of cerebral small vessel disease. And thirdly, they underwent a lot of memory tests. And the idea is that if methylglyoxal is bad, you would expect that people with a higher level of methylglyoxal in plasma would then have more signs of cerebral small vessel disease, as we can see on MRI. Also, if methylglyoxal would be bad, the higher concentrations would then result in worse memory function. But again, here, we didn't actually find very clear evidence for methylglyoxal from plasma being bad. We didn't find a lot of strong associations to suggest that there's something going on from the plasma. And this, together with like these animal studies that we've done and looked into plasma methylglyoxal, made us wonder, can methylglyoxal actually cross this blood-brain barrier? And again, this castle wall, can methylglyoxal go through the castle wall? And we found it quite complicated to actually look or measure this if we have the castle wall as a whole, if we have a whole system. So what we had to do is we had to resort back to a more simple system. You could say we kind of build our own wall. And it's of course not a real wall, but it's cells grown in a dish that look a lot like a wall. And what we did here, we just added MgO or methylglyoxal. And after a while, we just saw what would come through. And what we actually found is that, yes, there is some methylglyoxal coming through, but the amount is very, very small. Now, taking all this together, and let's get back to the brain. What we did is we actually investigated the effect of methylglyoxal on the brain, but more particularly on the brain microvessels. And we did this by testing this from plasma, methylglyoxal from the source of plasma, and methylglyoxal formed in the body from glucose. And what we saw is that we don't have a lot of evidence from methylglyoxal from plasma to have a negative effect, but we did see that methylglyoxal formed from glucose has a negative effect. In other words, the source matters. Now, why is this really relevant? Now, of course, there is a lot of research being done into how can we treat cerebral small vessel disease and neurological disorders, and for that it's important to show that the source of methylglyoxal is very important in your study design and the readouts that you're using. So this contributes to the knowledge of the scientific community. Moreover, if at some point, and we're not this far yet, if we want to find a treatment strategy for cerebral small vessel disease, it will be more relevant to actually look into the formation and regulating the formation of methylglyoxal or the detoxification, rather than actually targeting methylglyoxal in plasma. And with that, I hope I have been able to explain you that methylglyoxal affects the brain microcirculation, but that the source matters. And with that, I would like to give the word back to the prorector.
Thank you very much, Ms. Berends. Very well presented, clear presentation, and also well illustrated, and exactly on time. That should be 3 compliments in one. Before we start officially our corona over here, I would like to introduce also the members of your team. They have brought you in the last years, since 2020, I noticed, to this moment here. So the opposition team for the audience are sitting opposite of me, for those one. And I start from my side on the right side to the left side. So we have first Dr. Fouquier. He's assistant professor at the Institute for Cardiovascular Research here in Maastricht. We have a professor in the middle, Professor van Oosterbrugge. He is neurologist and the chair of the Department of Neurology. And the first promoter sitting over here is Professor Schalkwijk, and he's professor of experimental medicine, at our university. And by saying this, we're going to start, and I'm going to open the opposition, and the opposition will be opened by Professor Staals. And Professor Staals is professor in neurovascular diseases and also a neurologist at our university hospital. And I would like to give her the first word.
Thank you. Dear candidate, Mr. Berends, congratulations with finishing your thesis. I really liked the book. I really liked the little mouse. But I'm not an expert in mice, far from being an expert. I don't do any animal experiments. I'm a clinician. So I'm going to discuss with you chapter 5, the Maastricht study. It actually takes years to develop MRI manifestations of small vessel disease or cognitive decline, so I was not really surprised that you didn't find much association between plasma, MGO and MRI abnormalities or cognition. It was a cross-sectional study. But you end with that these findings need to be further confirmed in other studies. So if you had a chance to design a new study, how would you do this? Which design, which patient population, which outcome? How would you measure MGO?
Highly esteemed opponent, thank you very much for your kind words, and thank you very much for your question. So indeed, it's a cross-sectional study. If I would be able to design my own study, I think a follow-up study would be firstly of interest. Because in that way we can actually see if also changes in methylglyoxal over time, and we can, if that has an effect, and we can also get a bit more of an idea of causality. I think I also mentioned already some ideas. It would be nice to get more information of methylglyoxal changes during the day. So if I could design my own study, I would also include multiple measurements, multiple samples over the day and over the years to look into the possible causal effects on cerebral small vessel disease.
Mm-hmm. And what kind of population would you take? Because I think the disadvantage from the Maastricht study is that these people are rather, well, they are not too old, and they are...
Yeah. I think it would be interesting to use for this a population more from, let's say, the range of 60 to 80 or 90 years old, because from the age of 60, the chance or the risk of getting cerebral small vessel disease increases. So I think in this population, you would be able to kind of pick up the first effects, from my understanding. So I think this would be a more interesting population to look into, but indeed a slightly older one, because I think the youngest in this study were about 45 years of age, which is relatively young for neurological problems. Yeah.
And would you still measure plasma MGO?
Yeah, it's currently, I think, the best that we have. We cannot take any brain biopsies, of course, from these people to measure it in, because that would be the most interesting compartment. Cerebral spinal fluid would also be very interesting to look at, but it was found in a previous study that there is quite a strong association between the levels found in plasma and cerebral spinal fluid. So I think, considering the invasiveness of cerebral spinal fluid, I think plasma would still give a
that's
reasonable
true. Maybe
reflection.
CSF is not that feasible in a large study.
Also that, yes.
And what would you take as an outcome measure? Because you say I would do a follow-up study, but again, it will take years before you get cognitive decline and progression of white matter hyperintensities, for example. So is this the good design, a follow-up study and taking those clinical endpoints?
I do think that, for instance, white matter hyperintensities, because they are relatively common in the older population, perhaps you can look into the size, because now we use the cutoff value of, I think, a total volume of more than 3 milliliters, yes or no. Maybe looking into, for instance, the sizes that could give an indication. I do still think these markers on MRI would be interesting, but maybe look more into the functionality of the blood vessels, if that's possible. But I'm not completely sure on the MRI to what extent you can pick up small changes in vessel reactivity, for instance, in these participants.
So nowadays we are often using surrogate markers of small vessel disease, so not the clinical endpoints, and we can measure, for example, blood-brain barrier, permeability, we can measure neurovascular coupling. What would you take?
Based on my own findings, I would say the blood flow regulation would be very interesting. But I think the blood-brain barrier integrity would also be interesting, especially because it's also indicative of, or I think it's predictive also for lacunar infarcts, if I remember correctly. And since we did see some effect here on lacunar infarcts, or effect association, that perhaps that would also be of interest indeed.
Yeah. But in your other chapters, you didn't find an effect on the blood-brain barrier, right?
No, but we found an effect on the vascular response, so that would be interesting. But one of the things, we looked into quite large leakages of proteins, so extravasation of immunoglobulin, which are large proteins. So I think they would be more like looking into the extravasation of a smaller tracer would maybe give you a bit of an earlier sign of cerebral small vessel disease. Yeah.
Okay, I'm content. So we will take elderly patients. If it's feasible, we will do a CSF. We will measure BBB and neurovascular coupling. And you will take multiple time points to measure the MGO. I will say MGO because it's a difficult word. Yeah, right. Okay, so I'm content with this answer now. Give the word back.
Thank you very much. Then the opposition will be continued by Professor Tessier. He was a member of the assessment committee and is professor in analytical biochemistry at the University of Lille. And I also thank him that he joins today this ceremony. I give you the word.
Yeah, thank you. Thank you, Paul Rector. Dear candidate, it is the second time that I listen to some of your results, and it's always a pleasure. It's always very clear, very interesting, and I think you would need 2 hours to explain the brain, but we would need 2 hours to discuss that. We have a lot of questions, actually. But my first question is about intracellular formation of MGO. In chapter 4, but also in the discussion and then in the conclusion, you say that we found that increasing formation of MGO intracellularly does such and such effects. But actually, you never measured MGO in cells. So how can you say that the formation of MGO is intracellular if you cannot precisely measure it in cells?
Highly esteemed opponent, thank you for your kind words and thank you for your question. We actually measured methylglyoxal, not the formation, but at least the concentrations in the cells in chapter 6, and this is of course not in the brain. And from a lot of other studies, we have a lot of evidence that there is an increase in cells. I agree with you to say that indeed we did measure intracellular methylglyoxal concentrations in this study specifically for the blood-brain barrier. But considering the knowledge that we have on the source, I think it's quite presumable that it is formed in the endothelial cells predominantly, or the cells within the blood-brain barrier.
Because you also say that MGO measured in plasma is a reflection of the increased MGO formation in cells. But it's just a reflection. Is it a real causal relationship, or is it just a correlation between the two? What is in plasma do you think that what you measure in plasma in animals or in human can be the reflection of what is in cells?
I think it has some reflection of what is happening, but one of the things that we see, for instance, in chapter 4, with the from increased formation, we don't see actually a very large increase of methylglyoxal in plasma. So I think we do see an increase, so there is some reflection, but I think within limits. That in the extreme cases you might see it, but if there's like a short spike in increased formation within the cell, that this is something that you might not pick up in plasma. That it's a snapshot of what is happening in extreme cases. That's what I would think. Yes.
Okay, and you said that methylglyoxal is the suspect, is your suspect, but you also mentioned glucose, and the glucose concentration in plasma is at 5 millimolar, whereas methylglyoxal is only 250 nanomolar, so 20,000 times lower than glucose. So I know you would answer that MGO is more reactive, but still this huge difference of concentration would orientate my research in favor of glucose instead of methylglyoxal. What do you think?
Yeah, indeed, my first response would indeed be methylglyoxal is way more reactive than glucose. Of course, there's more processes that might occur as a result of hyperglycemia, and I don't think it's also only methylglyoxal in, for instance, diabetes that causes an effect. But I do think that because it's so reactive, and we know that it actually, we know how it can actually affect proteins by modification, also DNA modifications, that it would be a likely large player in this process.
Mm-hmm. And my third question is that at the beginning of your manuscript, you mentioned that the concentration of MGO in plasma is 250 nanomolars, and you mentioned a reference from the Maastricht study. And at the end of your thesis, you say that the concentration in plasma is between 350 and 450 nanomolars, and then you mentioned another study, the CODAM study. So I would like to know which one is the correct concentration of MGO.
I don't actually know by heart what the setup was of the CODAM study, but I do know that the values from the Maastricht study, they were in, it was like measured in an oral glucose tolerance test over time, and that's like the increase that you see after, I believe, an hour, or not. I would actually have to look this up exactly where it was, but I know that one of them was from an oral glucose tolerance test, and one of them was about fasting plasma glucose. So I think that's maybe the difference. Plus, so I'm also not 100% familiar with the CODAM study population.
Okay, because I was thinking, is it due to the population, or is it due to the analytical method to measure MGO?
I can actually see, because let me quickly check. The ranges that we have in chapter 5, because they are also from the Maastricht study.
Page 157, you have a conclusion about the CODAM study, I think, from 2015.
No, I'm currently looking into chapter 5, into the characteristics that we have of that study population, and there we also see a range from actually 250 nanomolar to 430 on average. So I think it's a difference in the fasting state plus the health of these participants.
Or maybe it's a range, and we cannot.
I mean, it's absolutely a range. It, of course, differs per person, age, disease, and what's going on. Also, maybe their last meal, even though they're fasted.
Has an influence on that. Okay. I have no more time.
There's time for a short question.
A short question. In the introduction, you mentioned the ratio, the plasma ratio between AGE and RAGE. I know it's not the main topic of your thesis, but I'm always curious about this ratio. What does it mean? Is it relevant to measure this ratio?
I think the ratio, because RAGE, we have RAGE in all different types of isoforms and locations, and we have the soluble, membrane-bound, and the effect is, of course, different in different cells. I do think that there are studies that suggest that the ratio might be predictive of disease outcome, for instance. I believe that there was studies done on the ratio and the effect on also I believe cerebral small vessel disease, that they do find an association. I'm not sure how relevant this is because there's a lot of different ligands, of course, for RAGE, a lot of different isoforms of RAGE. I think I also mentioned it here mainly because we're not really sure what the actual effect of RAGE is in the brain.
Okay.
Yeah.
Thank you very much.
Thank you very much. Then the opposition will be continued by Professor Jones. Professor Jones was also a member of your assessment committee, and she is involved in cardiovascular sciences and vascular mechanobiology at the University of the Catholic University in Leuven. And I'd also like to thank her that she joins us today. I'd like to give her the word.
Dear candidate, I'd like to start off by congratulating you on your thesis. You went from in vitro work to mouse work to a clinical study, and so the breadth of what you've covered in your thesis is very impressive. So I'd like to start off a question about your in vitro work. You used in vitro endothelial cells, brain microvascular endothelial cells, to study the BBB and to study this process in vitro. How well do they keep their phenotype when they're cultured in vitro? Do they keep the BBB? Till what passage can you use them, and when do they start de-differentiating from being brain capillaries?
Highly esteemed opponent, thank you for your kind words and thank you for your question. So it depends, of course, on the type of cell that you use. I know for the iPSC-derived endothelial cells that we use that once they're differentiated up to, I believe, passage 5, they keep their barrier properties quite well. And of course, this is not an entire blood-brain barrier model because you're missing so many different cell types. But with this, what the lab that I worked in also validated quite well is that up until about passage 5, you have a really good tight barrier that doesn't leave a lot of room for permeability.
Okay. And then in your title, you say the source matters, and you're talking about endothelial cells and parasites being the source of the MGO. But your knockout is a full knockout. And when you're feeding it systemically. Why is it that it's endothelial cells and parasites that are the source? And why not consider other cell types like, I don't know, perivascular fibroblasts or astroglial cells or things like that?
Yeah, so it's the hyperglycemic model then. So it's the cells that take up glucose insulin independently, and because of course the endothelium and the parasites are in closest touch with the circulation, that's why we think it's the most likely cells to form large amounts of methylglyoxal. But it's also immune cells definitely can play a role in this. Interestingly, though, we didn't find a lot of evidence for inflammation. It's not actually in the thesis. But because we didn't find a lot of signs of major inflammation, we think that it's not the immune cells in this case also that are able to produce a lot of methylglyoxal, and they do, that they're not the main source. And then we think that it's the cells closer to proximity. But this is, of course, something that you would need to test more up close. But it's difficult in a whole mouse system.
And I mean, age products are the endpoint of glucose use. Is the diabetes in your model affecting how much glucose, how much glycolysis versus how much oxphos is going on in endothelial cells? How does diabetes affect endothelial cell metabolism in terms of age production?
I don't think I fully understand your question.
Because it's an end product of glucose usage, is there a change with the diabetic model in the way that glucose is used by endothelial cells? Do you know if switch their metabolism from being purely glycolysis to start using oxidative phosphorylation, or?
I don't know based on my own, our own work. Based on literature, I think because the availability is very present, I would think that it's mainly glycolysis, also because there is an increase in methylglyoxal. But if there's any metabolic shift going on in the cells per se, I'm not completely, I wouldn't dare to answer that.
And then I was also wondering, so in situations like diabetes and whatnot, you have an increase in various things. And when I was reading this about MGO as an end product of glycolysis and whatnot, it reminded me very much in some ways of what happens with ROS, where lots of ROS is damaging, but a little bit of ROS is actually a signaling molecule.
Yeah.
Is a little bit of MGO a signaling molecule, and why do bacteria intentionally make it? What's, like, you talk about the pathophysiological role. What's the physiological role?
Yeah, so there is absolutely a lot of evidence for it having a physiological role as well. I think it's also discussed in one of the figures I show quite nicely, where during normal glycemia, you have the formation of methylglyoxal regulating glucose metabolism. So reducing, I believe, if I remember correctly, glucose transporters, also having effects on the gene expression, and in this way maybe serving somewhat as a feedback on glycolysis. And that if there is, for instance, diabetes hyperglycemia, that you get this overload of the system where also the binding of methylglyoxal to proteins becomes aspecific, and the binding to DNA becomes more aspecific, where we get disease. So there is a role indeed as a signaling molecule. And I don't know if I then answered your whole question.
The only other part was about what does it do with bacteria?
Oh yeah, bacteria. So also there are suggestions that it might be like antimicrobial, that it's part of the defense system of certain types of bacteria. There's also evidence of, for instance, I don't know if you've ever heard of Manuka honey. It's a whole health hype. It's a honey very high in methylglyoxal, and there's suggestions that topical application would also be antimicrobial. That the methylglyoxal from this honey would then, prevent infection. So the fact that bacteria produce it might be kind of their own defense system.
Okay. Do I have time for one more, or is it?
Yes, you have time for.
Okay. So my last question is, you saw in the mouse model with diabetes, you saw dysregulation of neurovascular coupling. How can you, but shear stress and flow can affect your BBB and whatnot. How can you differentiate the role of the flow from the role of the MGO itself? And did you look at cardiac function? Is cardiac function normal overall in these mice?
Cardiac function in this model or in this study specifically, we didn't look at, but we did look into blood pressure and arterial stiffness. Actually, one of my colleagues actually looked into this quite elaborately. And one of the things that we actually found is that there was no significant increase in blood pressure in these mice. So that's kind of one of the, that would be, for instance, a reason why you could also see a difference in effects. But we didn't find this. So yeah, there's of course this is a lot of the underlying mechanisms of what exactly is going on would need to be further investigated. But at least we see a slowing down of the neurovascular coupling response.
Okay, thank you very much.
Thank you very much. Then the opposition will be continued by Dr. Van Mierlo. He was also a member of your assessment committee, and he is involved as associate professor in the field of gliobiology, both at Maastricht University as well as Hasselt University. And I'd like to give him the word.
Thank you, Mr. Chairman. So I'd like to start off with congratulating the candidate and the promotion team with a very well-written thesis. I think you used state-of-the-art technologies, well-designed experiments to study your research question and to come to the conclusion that the source better. So congratulations with that. But I also have some questions, and I'd like to discuss with you first chapter 3. So in chapter 3, you provided 15 millimolar of MGO in drinking water to see if you could induce a cognitive phenotype. Is this concentration which you apply here, is this matching with what you could see in diabetic patients?
Esteemed opponent, thank you very much for your kind words, and also thank you very much for your question. We found a twofold increase. So relatively, this is indeed what we see in people with diabetes. Yes.
Yeah, I agree. I think it's well matching, but it brings me to another point. You're studying young animals, 8-week-old mice, and you provide this drinking water for 13 weeks, which is relatively seen long-term, but also short-term.
Yeah.
If we look into patients, diabetic adolescents only show cognitive symptoms after 10 to 20 years of being exposed to these concentrations. So if you would redesign your experiment, do you still stick to the 13 weeks treatment?
So we used the 13 weeks to make sure that also we wanted to see the isolated effect of methylglyoxal, and also if you go longer, you have then also an interaction with actual age. So aging and methylglyoxal together might actually worsen the cognitive function. So that's why we actually stuck with the 13 weeks in young mice. If I would redesign this study, perhaps I would go for the endogenous formation of methylglyoxal, but if we want to look into the methylglyoxal source of plasma, I think it would be more interesting to not necessarily look into like extending the duration, but probably combining it with an existing mouse model for neurological problems.
Yeah. And how would you go about using older animals? Because you also nicely describe in your discussion, in your general discussion, that there are epigenetic imprints being generated, especially in aged phenotypes.
Yeah.
Would that be an option?
I think aged mice would also be interesting, indeed. Although there's also already some studies that show that there's also not a lot of effect on methylglyoxal and plasma in aged mice, although they didn't look into the cognitive function. Yeah, I think we might see more effects than what we do in young mice, but I don't know how much we would see also, yeah, in aged mice, if the cognitive phenotype would be strong enough to pick up.
Yeah, and also when I was reading this chapter, I was wondering, could you have missed the effect? Because you indeed use several experimental tasks to assess cognitive function in these animals, but for instance, the object location task, you use a 1-hour inter-trial interval between trial 1 and trial 2. But if we look into Alzheimer mice, you only find an effect on the 5-hour inter-trial interval, so this might be too short. Do you have any comments on that or reconsiderations on how to approach this?
Yeah, I think it would indeed be because we didn't know what we would expect when we started this and when we designed the study. I think indeed maybe using more sensitive methods for cognitive function, the chance of picking something up would of course be bigger. But because we don't see any evidence for damage of the blood-brain barrier or neurovascular coupling response, inflammation, we don't have any other evidence that supports that anything might be going on. So I think, yeah, you could use more sensitive tests, but I doubt we would pick up something there.
Okay, thank you. This also brings me to my next question, because in chapter 4 you use a diabetic model, and the same tests are being applied. So my question also relates to that chapter. And in that chapter, you only find an effect, a small effect, on the Barnes maze, and you only analyze day 2 till 4. So why did you take out day 1 of the analysis?
We took out day 1 because there's a lot of variability, and the idea was that from day 2 they should start like really remembering, and that's where we could pick up the differences. And especially, on day 1, there's just a lot of variability that would not really pick up clearly what's going on the days after. Yeah.
Okay, but in this context, it might be useful to look into a learning curve in the object location task and not stick to the 1-hour interval, right? Can you agree on that?
The learning curve of the object location task.
So increasing the inter-trial interval till you find the subtle difference on which you find the cognitive drop.
Yeah, that indeed would be interesting. Yeah.
And when looking at the experimental design of this 4th chapter, you start off with elevated zero maze in your behavioral testing, and this is an anxiety-induced assessment of of the cognitive impact on their anxiety feelings. And it's known that this also triggers the production of corticosterone in these animals, and this in turn has an impact on the tasks which you perform after that. So what made you choose to start off with the elevated seromase?
It's a great suggestion to maybe next time do this at the end rather than the beginning. We didn't actually choose, or at least I don't remember we chose a particular order for these tests, at least not for as far as I'm aware of, to be honest. So I'm sorry I can't answer that question.
Nevertheless, you see an effect on the Berends maze, so it's still there, but there might be an interference, perhaps in one direction or the other, right? Yeah. Okay. Then I have a short last question. So in chapter 6, you studied the intravenous injection of MGO to see how it impacts and reaches the brain or not. And I was wondering, and then I was looking up the half-life of MGO, and it's actually quite short. It's 30 seconds or a bit more. By looking only after 4 hours, might you have missed the impact? Is it already metabolized?
So also indeed in the plasma itself after injection, it is not detectable or increasingly detectable anymore after 10 minutes, more or less, I think, or even shorter. So the idea with this was just to see, can we pick it up over time? And well, there's a lot of limitations to measuring it that way, because we measure the whole brain parenchyma, not just the blood, the blood-brain barrier. And that's why we resorted to this in vitro model. So I think, we might miss it, but we can't tell. Maybe if you would repeat it with labeled methylglyoxal, we'd have a better idea. But also because the blood-brain barrier is not that big of it's quite a small fraction of the whole brain parenchyma. We tried and we saw nothing, and that's why we resorted to an in vitro model. Yeah.
I agree. I think the labeled MGO would be a very good idea, but I also realize it's challenging, and there are other challenges in that perspective. So thank you very much. I'm satisfied with your answers, and I'll give back the word to the prorector.
Thank you very much. Then the opposition will be continued by Dr. Wiesmann. He was also a member of your assessment committee, and he is involved as assistant professor at the Department of Medical Imaging and Anatomy at the Radboud University Medical Center in Nijmegen. And I also like to thank him that he joins today. And I give you the word.
Thank you very much, Rektor. The candidate and the promotion team, congratulations on this nice thesis. And I was really also liking your critical comment on the journal policies. So, but luckily, one of the papers is now really also published. I think you were struggling with. So I really like that comment. We also had in the last conference really a session on it, and that's why I know the problems with that kind of issues. And continuing also with chapter 3 and also on Dr. Van Mierlo's comments, I was really interested, how did you overcome habituation effects in all these behavioral tests? Because you repeated them at least, let's say all of them 3 times. And how did you counteract habituation effects? How did you, for example, in the Berends maze, counteract these effects?
Esteemed opponent, thank you very much for your kind words, and thank you for your question. So with habituation effects, do you mean the habituation to the arena as because they're repeatedly measured, or the habituation to the arena before we start the measurement of the testing?
This too, and also the habituation to the test, because you have really relatively young animals, and of course if you repeat these kind of tests, they know how the test works, and that's also slightly reflected in the results you showed in the learning curves, which are getting steeper and steeper. Yeah.
So there was quite a bit of time between the behavioral testing, and so far, at least in the data, the main effect that we saw was in the Berends maze. That indeed the second time they knew pretty much, or it seemed that they knew how the task worked, and that's why we also, one of the reasons that we only performed this one twice. But yeah, we kind of assumed that with the time that we had in between, they wouldn't remember in such detail that it would affect our test. That was our assumption in this.
And I guess you use a different kind of rescue hole in your Barnes maze, or did you use the same position?
No, different
Okay,
one.
good,
Yeah,
good. So,
absolutely. Yeah.
And but have you also checked if they remembered on long term the old position of the former rescue hole, let's say, in the Barnes maze?
We didn't test that,
Okay,
but that would be interesting.
So
yeah.
more on short term now. Okay. And then continuing also again on the continuous comparison over time, if you look at the figures on page 74 of the systolic blood pressure, you show continuous lines, while you show, for example, on page 66 in the different kind of behavioral tests, you show more dotted figures without continuous lines in between. So I was wondering, how were you performing the statistics? So when having multiple time points, were you also looking what is happening from, let's say, baseline towards the next test time point and then follow up? Or were you really only looking at each time point?
We did a two-way ANOVA for at least like I think both of, we did, two-way ANOVA for all of these. So we also looked in effect over time, and we thereby also corrected for multiple testing. And then whether we compared for instance, the first measurements versus the last measurements. Yes. I believe we did test for this, but I would have to...
Because I saw none of these effects in your figures back, so that's why I was asking, because I was curious on the systolic blood pressure effect, for example, from 7 to 14 weeks, where you see an increase in the MGO group, the delta is really going up, so almost up to pre-hypertensive state. So I was wondering if you could figure it out, because on baseline, your groups were both control groups, right? So so that's maybe an extra multiple comparison you added, which is also masking your effects, because like you said, you used the multiple comparison for all time points, and so here you have always two groups. So maybe you overcorrected your data. So it's good, but maybe you're masking a subtle effects.
Yeah, I believe that we saw. There wasn't. We did see an effect of. I believe time and treatment. I would have to. We. It's in C. So we saw an effect of time, but not interaction.
Okay.
I think would. Yeah. And then the multiple testing didn't show anything significant. So I think it's maybe there is. I think maybe if we increase the statistical power, for instance, we would see a statistically significant effect.
Yes.
Yeah.
And then continue again on the same page with the water intake. How did you measure the water intake? Was it measured per kg or individually measured? Because you state also in the follow-up chapter that it's really important, the water intake, especially in diabetic mice, which drink double as much as normal mice.
Yeah. These were indeed measured per cage, and I think in the study that we used here, the mice were housed individually for the measurements of the water. So it's indeed we have the water intake points per mouse, which is per cage.
And how many cage were you housing? How many mice per cage were you housing?
8 per group, so 8 cages.
So you had 8 mice per cage? No.
No, 8 cages.
Yes.
So 8 mice, 1 mouse per cage.
Oh, one was also solitary house.
In this the measurements that we got, the water intake and the body weight, and they were individually housed, if I remember correctly.
Okay. Because comparing the results on page 74 to your results in chapter 4, on page 94, there you show that the control mice almost drink half as much as in your chapter 3, where you also have younger control mice. So I was curious on if you perform different kind of measurements.
They were housed socially there.
Okay.
So there were about, I think, 2 to 4 mice per cage, in combination with... because we then correct the water intake per cage to the body weight of the mice. And in this case, it would be then 4 more mice, so more body weight, and 1 bottle leaks a little less than 4 bottles. So I think in this correction, this is maybe where the discrepancy comes from.
Do you have time for a short question?
Unfortunately not.
Okay, thank you.
So we have to proceed. So we move Thank you very much. We move to our last opponent, and we also go now from on-site to online, and we welcome Professor Martinez. She is professor in neuroinflammation and neuropsychiatric disorders at our university, and I'd like to give her the word.
Thank you, dear Pro-Rector, dear candidate. First of all, congratulations for the very nice thesis. A lot of work that spans from working with animals, in vitro studies, and also working with human samples. I think that's a level of complexity. Also, my compliments to the supervision team. My questions goes to chapter 3 and a little bit elaborating on what Professor Van Mierlo was asking before. So I am very interested about your study, and I really wonder whether you really satisfy in your conclusions when you conclude that you don't see any effect of increased MGO. And I would like to elaborate. So you, when looking to your analysis, look to several parameters, including inflammation and gene expression. Do you miss anything else that you could have analyzed regarding gene expression, since you only focus on the glucose metabolism pathway?
Highly esteemed opponent.
Could you elaborate about that?
Yeah, highly esteemed opponent. Thank you very much for your kind words, and thank you for your question. So if I understand correctly, you're wondering if we maybe would have done more gene expression analysis, that we would have maybe picked something up, or am I misunderstanding this question?
Well, you really focus in the glucose metabolism pathway as the one to be altered or genes, but you don't look to a broad pathway analysis. So you might have missed effects in other pathways in your study. Do you agree about that?
Yeah, of course, there's always things that we can miss. Considering that we didn't find any with the things that we have investigated, we didn't find any evidence. So I think any pathway analysis in this would have, it's a bit of a choice of whether it's worth further diving into. We maybe could have missed something, but considering we didn't find a lot of effects, I'm sure.
I would like to disagree on that because, for example, you also mentioned MGO can be also produced via degradation of DNA and proteins, so via this metabolism, not only from glucose metabolism. So I was wondering whether you really, the design of your study might have been adequate to study effects of NGO, or whether perhaps NGO that is degraded from other pathways might have having a higher impact in the brain. For example, the lipid metabolism.
So is it the idea to look more at a gene regulation expression? I don't think I fully understand your question.
My question says, so if you would have looked to other indirect effects of your analysis, so instead of looking to the direct effects in the typical pathway that might be affected, if you would have looked to other pathways, if perhaps you might have picked up any MGO differences or effects on the animals, that's my question. Or if you have any other idea on how you could have designed that experiment to have perhaps seen more successful on observing an effect of MGO.
And then are you referring now to chapter 3 or chapter 4?
Yes, chapter 3. I am just referring to chapter 3.
Yeah. So whether other sources of MGO might then have an effect, or
if you think that changing the way you administer MGO or looking to other pathways besides the one you have analyzed might have given you a different result.
So for instance, you mean like instead of supplying it through the drinking water, that if injection or other administration might have a different effect. Yeah.
For example, that's one thing you can elaborate about. Another thing is whether the downstream analysis that you did was perhaps revealing everything on this study or not.
So firstly, I think if we would administer this in a different way, that there is evidence that shows that if there's repeated injection, that there might be a different effect, but it's of course a more invasive and less consistent administration. I do not exclude the possibility that we actually missed any effects of methylglyoxal, but also the way that we conclude this study is that at least methylglyoxal from the drinking water, if otherwise healthy, that we don't see any effect on the brain microvasculature and the brain in at least the parameters that we looked into. And that in combination with another disease or another model, another administration pathway, that maybe you can pick something up. And it could still be the case that we have missed something.
Okay, well, if you look now to figure 3.3 in your chapter 3, where you elaborate about or measure IgG extravasation. So you are measuring whether IgG is present or leak through the vessels. Then I wonder if you have missed information in this figure, if you look to the figure and you then point out that is part K in the figure, and at the magnification of your microscopy, it could have been possible that the diffusion of IgG was missed. Could you elaborate about that? If you will be repeating this analysis, will you have measured the IgG or the leakage of IgG via immunofluorescence? So will you have used other methods? I'm very interested how you cut your sections for doing this analysis.
Yeah, so indeed we only looked into IgG, and I think in another study it would have been interesting to look into also smaller proteins or tracer extravasation. And we indeed use sections, so it could be the case that you miss extravasation in other parts of the brain. And of course, if you would do tissue clearing, maybe, I'm not completely sure about immunoglobulin, if that's possible.
Dear Ms. Candidate, you're allowed to finish your answer, but you don't have to. You also can stop, whatever you prefer.
So I would like to still say that I think indeed we could have missed something here, but because we did the same for all the mice, I think it's still safe to say that the statistics are reasonable.
Thank you very much. Dear Ms. Berends, dear Ms. Candidate, the time appointed for defending your thesis has now passed. The degree committee will now withdraw to discuss the quality of your thesis, but in particular the way you have defended your thesis this morning. I request that you and your company await the results of our deliberations in this room. Thank you very much.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side. 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. On the line, I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Get the mileage, traveling so far from home. Gathering knowledge, other connections. future that we go for. And even though these nights seem long, I know that I am never on my own. Know you've won the battle once you've grown. Long road to the south side, 10 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side, no place like home. If it's questions that you've got, go the extra mile and not. Long road to the south side, 10 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side, no place like home. If it's questions that you've got, go the extra mile and not. What's on the line? I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. 'Cause I'll go the extra mile.
Dear Ms. Berends, the degree committee here present has assessed the quality of your thesis and in particular the way you have defended your thesis this morning. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you officially the degree of doctor. Professor Schalkwijk is authorized to confer you this academic distinction in accordance with Dutch university customs, and therefore I invite your supervisor now to take officially the floor.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Eline Berends, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, by the secretary, and the other members of the committee, and fixed with the official seal of the university.
I will give now the word to Professor Fouquier, or Dr. Fouquier, who speaks out the laudatio.
First of all, of course, also for me, Eline, my congratulations. You did very well. I'm really proud of what you have achieved, actually, within 4 years. It's really great. And indeed, the honor for the laudatio is for your daily supervisor, Sebastian Fouquier.
Thank you, Professor. All right. Dear Dr. Berends, dear Eline, it is an honor to be among the first to congratulate you on this tremendous academic achievement. And on behalf of the entire supervision team, I would like to take a moment to reflect on your PhD journey, a journey filled with not only academic success, but so much more. And how did it all begin? When we were looking for a candidate for this new project, Irina, I think she's online, a PhD student in the group of Professor Trinchas here in Amsterdam, who I had supervised during her master, spoke very highly of you. And we knew we could trust this recommendation, and in March 2020, you joined us right at the start of the COVID-19 pandemic. Our first meetings were online and during the lockdown, so it made it quite an unusual start. You joined both the Department of Internal Medicine, under the group of Professor Casper Schalkwijk, and my group at the Department of Pharmacology and Toxicology. And right from the beginning, you immersed yourself in the project, and over the past 4.5 years, you've accomplished so much. As demonstrated in your thesis and discussed, you've worked on a wide range of studies, from in vitro to in vivo experiments, and to analyzing also large epidemiological datasets. You also proactively learned and developed new methods and tools. And over those 4 years, you co-supervised at least 7 bachelor's and master's students. Tried to retrieve all the names: Lena, Naima, Ines, Wesley, Julian, Isa, Lisa, just to name a few. You presented at countless local and national and international conferences, both with posters and oral presentation. And you have even been selected to compete at the Dutch Medicine Days taking place next week. You have authored 4 papers, 2 of which are already published, while 2 others are under revision or submitted, and another one is also in preparation. This significant body of work has contributed greatly to your PhD thesis. Beyond these very impressive accomplishments, you have shown a tremendous determination. You looked out for the best possible training and adhered to a very strict schedule. You applied and you were selected for the prestigious Carole course in Bordeaux, for example, where you gained cutting-edge skills and met leaders in the field. You also received the prestigious Harry Stryker BAFTA Fellowship, allowing you to train in Professor Britta Engelhardt's lab in Bern, an opportunity that not only contributed to your chapter and your thesis, but will likely also result in another publication. So also a big thank you to Professor Engelhardt, and I think she would be also happy with the castle on the hill. As I mentioned, you also stuck to a very strict timeline, compiling, writing, and completing a PhD thesis with precision, and you made it seem very effortless. Aside from your scientific achievements, you've also made a remarkable impact on those around you. You actively contributed to society and your peers by joining the I'm Karim PhD Student Association, participating in the Pint of Science, and helping also to organize some running activities for Karim. You have been a crucial social bond for our research institute and our respective groups, which we deeply appreciated. So what now? What's next? While I've listed many of your accomplishments, what truly stands out is your mindset. It's not just about what you have done, but how you approached every challenge with determination and focus. As promised, I would like to say a few words in the native language of the candidate, so Dutch it is for me. But instead of the usual réfléchitaire, I would like to quote a French philosopher. Yes, a French philosopher. He lived in the Netherlands and spoke Dutch fluently. So that means also that's possible for me. René Descartes. René Descartes was a French philosopher, a mathematician, and a scientist. And he once wrote, "Kennis is macht, maar karakter is meer." Knowledge is power, but character is more. And this perfectly reflects your journey. While your intellect and skills are beyond question, it is your incredible determination, integrity, and strength of character that have truly shaped your success. So today, we celebrate not only your academic accomplishments, but especially the remarkable person you've become. We are so proud of you, but now it's time to let you go. I'm very glad you have secured a position in the renowned group of Professor Helena Karlström at the Karolinska Institute, and I'm certain you will continue to thrive and grow there, and I see a very bright academic future ahead of you. Since you are staying in the field of vascular dementia, I'm sure we'll cross paths at future conferences. But remember the Limburg saying, the life is always better in the south. So you're always welcome back in Maastricht, and our doors will always be open for you. Dit is geen afscheid, maar een tot de volgende keer. Sorry. I will sincerely miss you, Dr. Berends. Congratulations. And I give the word back to the porter.
So thank you very much. And I would like to start to congratulate the new young doctor, Dr. Berends. Feels good?
Yeah.
Yes? Okay. Also on behalf of Maastricht University, I would like to congratulate you. I would like to congratulate also the team which helped you in the last 4 years to get to here, to this point. I would like also to thank the members of the Corona who took the time on site or online to be here and to ask some questions to you. Maybe it was some tough questions. I should also mention that the entire Corona was impressed about your presentation. It was a very good presentation, and it was the one which we as university exactly liked. It should be for a lay public, and it should not be a presentation at a scientific meeting. So I think that also your family in the first row, which I'm going to congratulate also now, that they understand your presentation and they know a little bit what you have done. So I like to congratulate them as well. And by saying this, I would like to close the ceremony. Therefore, I use my hammer, which you haven't seen, but it's right here. I close the ceremony, and I would like to mention the following for the procedure. In the procedure, we're going to make some pictures. First, we make one picture here in the audience with our online Professor Martinez. Then we make an additional picture on the stairs, but you in the audience don't have to wait for this. There will be a reception, which is in the Refter, which is here, right here, if you go out of our aula to the right. And there's already coffee served, tea, and whatever the candidate has chosen. I'm not sure what that's going to be, but you can already decide there, and we follow you there in a couple of minutes later. Thank you very much.